about parasites, you might think of words like disease or infection or maybe deworming your dog, but they're actually very useful in studying ecology. My name is Veronica Torres, and this summer I worked with the Parasite Ecology Lab Group to study, as Sammy said, the diversity and burden of parasites in this species of fish. Now, the way that we use parasites to study ecology is through systems like this. As you can see here, we have a tapeworm. It enters this host, and then when this host is eaten by a fish, now the tapeworm is in this fish is eaten by a larger fish, the tapeworm is here, the tapeworm lays eggs, and the cycle continues. In the case of the fish that I'm looking at, sometimes it's this intermediate host, sometimes it's the final one, but in all cases, parasites will only use links like this that are strongly established. They're not going to enter a prey item that's not going to be consumed by the final host, because then the parasite can't complete its life cycle. So keeping systems like this in mind, where parasites go from predator to prey, there are a couple of other factors to remember when looking at systems like this in fish. One of the factors that affects parasite accumulation is size. A larger fish will have more parasites than a smaller fish. In general, larger fish tend to be older, and as they've gotten older, they have had more time to accumulate parasites. Parasites are represented here by these beautiful squiggly lines. And trophic level, which is just where the fish lies on the food chain. If this large fish were to eat this smaller one, it would then get the parasites of that smaller fish. So size and place on the food chain both influence what kind of parasites and how many parasites that fish would have. In the case of the fish that I'm looking at, I chose it because it's abundant in kelp forests right off our coast, which makes it easy to get samples of and to study. It's part of a larger kelp forest food web project in which we look at the parasites of many animals in the kelp forest, and using parasites and those predator-prey relationships we discussed, we can see how all those kelp forest animals are connected. And little is known about the parasites of this species of fish, so any data that this project gathers would be potentially interesting. And the last interesting factor is that this fish actually changes sex from female to male over the course of its lifetime, which could potentially affect what kind or how many parasites it has. So it could be that females and males have the same parasites, the same number of parasites, and there's no difference, or they could be seen as two distinct categories in which we see differences that may be related to the fact that they change sex. To answer this question, if parasite assemblage, number and type of parasites differs between the sexes, keep in mind their size. If one sex is larger, perhaps that influences what kind of parasites they have. Or if they eat different things, we might see differences in their parasites as well. So in order to answer this question, the first thing I had to do was perform tissue swashes, in which I took the soft tissues of all of the fish that I looked at and made them very thin in order to look under a microscope to locate parasites. And I identified each of those parasites that I found. This gave me both a total count of the number of parasites in a fish, which we call the parasite burden, and an idea of how many types of parasites were in each fish, which we call the parasite diversity. As I mentioned, diet is an important factor in influencing what kind of parasites a fish has, so I also looked at the gut contents. But as you can see, this picture here, gut contents get degraded very easily by stomach acid, and they're only a temporary snapshot of what the fish has recently eaten. So while I could identify several items in the gut contents, which gave me a metric for diet diversity, which is how many species the fish is eating, this was more to supplement the parasite data. As I said, parasites accumulate over a fish's lifetime. So while this is temporary, parasites give us an idea of the fish's behavior over its whole life. So using this data, the first question that I attempted to answer was, is there a difference between the parasites of female and male rock grasses? Here on the x-axis, we have the sexes, female, male, and then the transitional stage between female and male. And here is parasite burden, which, as I mentioned, is the total number of parasites. Though our sample size was small and did not include very many females, we can at least say that for our sample, females did have a slightly higher parasite burden. But when looking at diversity, we did not see that pattern. Here you have sex, again, on the x-axis, and parasite diversity, which is the number of parasite types. And all three sexes had similar diversity counts. As I mentioned, size is one of the deciding factors in the parasite assemblage of fishes, so I decided to compare the size between the sexes. here you can see males, females, and the transitionals again. But all the medians are very similar, and although the males have a higher minimum and maximum, which was not able to be calculated due to the small sample sizes of females and transitionals, all of these fish fell within the size range of males, which led me to eliminate size as a factor that would be influencing any potential sex differences. Because size was a difference between the sexes, I decided to compare size of all of the fish. Here we have length, since that's more standard than weight, which can fluctuate based on how much a fish has eaten or if it's a female carrying a lot of eggs. So I compared length to parasite burden, and all of these dots are color-coded by sex, blue for male, orange for female, and gray for transitional, to see if all sexes followed any patterns that we saw emerging when comparing length to burden. You can see there's a peak in mid-sized wrasses here, around 170 millimeters, and that peak is more obvious when we look at length versus parasite diversity. Now, since this was such an interesting factor in terms of all the fish, I'll discuss it later in comparison to this next pattern, which is diet diversity versus parasite burden. While you don't see a pattern emerging in relation to burden and diversity here, you do see a pattern of a positive trend when comparing diet diversity to parasite diversity. Now, since both diet diversity and length were positively correlated with parasite diversity, especially for those mid-sized wrasses, that led me to this next comparison, where I compared the factors of length and diet diversity, both related to parasite diversity. And though no clear pattern emerged, the fact that all three factors are linked is still potentially very interesting, and I will discuss it later, especially in terms of future work. So, my main question that we started with was, does parasite assemblage differ between sexes? We did find that for females, they had a slightly higher parasite burden, at least for our sample, but there was no difference in diversity between female and male rock wrasses. Now, why would this happen? One reason could be that females are eating more because they are actually making eggs, which is something very energy intensive and which we know requires lots of energy in fish and many types of animals to make. While we weren't able to get data on how much each fish was eating over the course of its lifetime, as I said, parasites show us who's eating who in that cycle of parasite transfer, and gut contents are only a snapshot. I don't have a way to verify this yet, but this is my current hypothesis that if females are eating more because they need to make eggs, they're exposing themselves to more parasites, which we see reflected in an increased burden. As for the lack of difference in parasite diversity, we see that females and males were generally eating the same types of food, which could mean they're generally exposing themselves to the same types of parasites.